Hello everyone. So today we are going to be doing reproductive physiology and also embryology in lab today, but as you know, that didn't happen. So we're going to do it virtual style instead. So starting with slide number 1, I want you to remember that meiosis, let's underline that. Oh, I don't get to? Okay, fine. All right, I guess I'm just going to use my laser pointer instead. So meiosis is the way that we divide our DNA so we can combine it with the DNA of somebody else. And meiosis only happens in gametes, it doesn't happen anywhere else. And so one term we need to keep track of is haploid. So a haploid cell has half of the genetic information of a diploid somatic cell. So the cell types of your body can be divided broadly into two kinds, your somatic cells or somites, and those are the cells of your body that make up the bulk of you, all the cells we've been learning about so far in class. Your haploid cells are the ones you've just been introduced to, the gametes, and these have half of the genetic information of a somatic cell. Gametogenesis is the process by which a mature haploid gamete is formed. Homologous chromosomes are chromosomes with the same genes on them, but not necessarily the same alleles of those genes. So an allele is a variant of a gene for a particular trait that differs from another allele. So for example, some alleles are dominant and others are recessive, and the combination of all the alleles that you have determines your phenotype, or what you look like. A zygote is the product of an egg meeting a sperm, so it's the first cell of a new individual. This should say diploid, not diplod. So diploid is the opposite of haploid. So diploid cells have two sets of genetic information, pairs of chromosomes, and those pairs, within a pair, you get one chromosome from your mom and one from your dad. Mitosis is cell division of diploid cells. And meiosis 1 and 2 are the final steps to prepare a gamete for combination with another. Also important to note that gametes come from stem cells. In one of the sexes, the stem cells stick around so that you can continue producing gametes well into old age, and in the other sex, the stem cells are differentiated by the time of birth and you don't get any more. Let's see if I can pen. Yay, there we go. Okay, cool. So for some reason my pen wasn't working earlier. Okay, so synapsis is when pairs of homologous chromosomes, remember homologous means same genes, different alleles, cozy up to each other, and that allows them to form tetrads, which is two pairs of chromosomes. Crossing over is when their little legs or arms cross, and that is how genetic information is exchanged. So the reason that you don't look exactly like your brother or sister if you have a sibling is because although you share 50% genetic identity, you don't have exactly the same parts of your mom's and dad's chromosomes as your siblings, because the crossing over is random and the assortment of the results of crossing over is likewise random. So there's genetic reshuffling that happens in there. Dyads are the two halves of the pairs that are actually exchanging. And you need to know just generally when DNA replication occurs over the next couple steps. So let's look at those. So prophase 1 is named for the events, and the fact that it is 1 indicates that this is part of meiosis 1. So there's two phases of meiosis, phase 1 or meiosis 1 and phase 2 or meiosis 2, and each of them have phases that resemble mitosis but are not mitosis. So each duplicated chromosome pairs with its homologue. So duplicated means there's two arms that are the same, and homologous means that this chromosome here, this like lighter aqua one, has all the same genes as the blue one, but not the same alleles necessarily. So these guys form a tetrad, and they cross over. So that's what's happening right there. Each chromosome then becomes attached to the spindle, which, remember, the spindle forms from the centrioles and the microtubules that radiate out from the centrioles, which are here and here. And so those are going to form the spindle array once the nuclear envelope completely goes away. So you can see here the nuclear envelope is degrading. So here we have a very classic metaphase appearance. There is a metaphase plate, which is right here, and the lemon-shaped spindle with centrioles at each side. So the chromosomes are pushed and pulled into the middle of the cell, and that completes the spindle. And of course, the kinetochore is attached to the spindle fibers, and these are going to pull the chromatids apart. So in anaphase 1, homologous chromosomes segregate. So these spindle fibers are getting longer to push the two sets of chromosomes away, and the spindle fibers are getting shorter to pull at the same time. But note, in each case, the sister chromatids remain attached. So this chromatid and this chromatid aren't separate yet, so we still have a full X-shaped chromosome. That won't be true for long. Telophase 1 looks a lot like regular telophase, so the chromosomes are moving towards the poles, and the nuclear envelope is going to reform. And this is usually followed by cytoplasmic division, which you can see here. Microtubules attached to the kinetochore again in prophase 2. And now, instead of forming a tetrad and crossing over, these individual chromosomes are going to be pulled in half, and that's going to turn the cell from diploid to haploid in finality. So, duplicated chromosomes again line up at the spindle, midway between the poles. And in anaphase, this time the sister chromatids are separated. So now the duplicated chromosome is no more, and we've got one chromatid moving to one end and one chromatid moving to another. And remember, these two sister chromatids have the same genes on them, but different alleles. So for example, this one has a little foot that is the result of recombination. During telophase 2, the chromosomes arrive at opposite ends of the cell, and the nuclear envelope forms around each set of chromosomes, and the end result is 4 haploid cells. So note, we have from an initial tetrad of 4 sister chromatids, we've got relatively unchanged in the other direction, and then 2 that have recombined and crossed over. Okay, so this picture is showing you a little wedge or pie shape view of the edge of a seminiferous tubule. So each of the tubules that you will see has the following stuff going on inside of it. So this is the edge of the tube here. And then down here where the sperm are, this is the lumen of the tube. So you can orient yourself using this picture. Okay. Now, first let's address what this yellow stuff is. So this is the cytoplasm of sustentacular cells. And sustentacular cells are really, really big sheet-like cells, and the developing sperm are kind of nestled between them, like between the pages of a book. And it provides a safe, nourishing environment for the spermatogonia to differentiate into spermatids. So the spermatogonium is the stem cell. And after puberty hits, follicle-stimulating hormone stimulates the division of the spermatogonia to form one additional spermatogonium. So each stem cell division forms one stem cell and one type B spermatogonium, which is the cell that is destined to differentiate into what will eventually be 4 spermatozoa. So note that there's different numbers of N's here. 2N equals diploid, and 1N equals haploid. So what that means is that meiosis is taking place as you move towards the tube. So there's a directionality to this as well. The closer you go to the lumen of the tubule, the more of meiosis will have been completed. So as primary spermatocyte is still diploid, after meiosis 1 is completed, we get a haploid cell, and then each of these becomes a product of 1, 2, 1, 2, 2 additional haploid cells for a total of 4 spermatids from one cell. So each stem cell gives rise to 4 spermatids and an additional stem cell. So the direction of meiosis goes this way. Now, spermatozoa, once they are fully formed, and they look a lot different from the little spherical guys they used to be, these are going to be moved into the reedy testis and ultimately into the epididymis, and that is so that they can mature and learn to swim and get ready to go off and possibly do their job. So the part that we were looking at in the previous slide was more or less a section that looks like that. So we can see each seminiferous tubule section contains numerous spermatogonia and some cells undergoing various stages of meiosis. And then once you get to the lumen, we've got spermatids with their heads embedded in the wall and their little tails poking out into the lumen. Also note that the Leydig cell, these produce testosterone, is not inside of the tubule. They are outside. So if I ask you on a test, true or false, the cells of Leydig are inside the seminiferous tubules, you will say no. Also note that Sertoli cell is another word for sustentacular cell. Okay, so here's what this looks like in histology. And indeed, you can actually see the little tails of the spermatozoans sticking into the lumen. So if you know that development of sperm and spermatogenesis occurs from the edge of the tube to the lumen, you can use that as a spatial cue to kind of figure out what you're looking at. So at the very edge, these guys with the lighter colored nucleus are going to be spermatogonia. And just inside, there's going to be larger cells with a larger, darker nucleus, and these are going to be primary spermatocytes. So here's a primary spermatocyte, here's one, here's one, here's one, here's one. These little guys with a smaller nucleus and a dark nucleolus are going to be secondary spermatocytes. And then these little guys here with a dark, more oval head here, these are going to be spermatids. You can also see often very triangular patches of Leydig cells, and that's just a consequence of 3 tubes coming together to make a triangular space. They're not always triangles, but oftentimes they are. All right, so here's another view of the same. We've got the close-up view of a spermatid. So this is having undergone meiosis completely. Now it's going to differentiate into the classic sperm shape because this thing does not look much like a sperm, doesn't have motility, doesn't have a flagellum. So spermiogenesis, which is not the same as spermatogenesis, spermiogenesis is the process by which a spermatid becomes a spermatozoan, and that suffix zoan means animal, and that's because, of course, sperm swim, and when they were first observed, someone thought, oh, hey, they look like little tiny animals. So this is how that works. The acrosome forms from an acrosomal vesicle. So it's a specialized lysosome, basically, that's full of digestive enzymes that digest a hole in the zona pellucida of the egg. And that's going to become more conical along with the nucleus. The flagellum will begin to grow, and the microtubule rotor shaft will form. Mitochondria are going to arrange themselves around the midpiece, which is the area that twirls the flagellum, and then the mature product has a head, a midpiece, and a tail. Here's a closer up view. So there is this structure that is long and tail-like called the flagellum, and this whips around in a spiral-shaped pattern to propel the sperm forward. And this is powered by the ATP from all of these mitochondria and rotating centrioles. So there's a centriole, proximal and distal centriole shaft, and these have motor proteins that rotate them in a circle, and that's how the flagellum works. So that's how swimming is accomplished. Now, oogenesis, and it's not oogenesis, it's oogenesis. So you kind of prolong the O or almost do a glottal stop and make it sound like you're saying two of the O sound in a row. This is a little bit different because the stem cell the stem cell progression is not the same. So in males, each division of a spermatogonium produces one additional spermatogonium and a future set of 4 spermatozoans. In females, during fetal development, meiosis 1 begins, but it arrests in prophase. So we have an oogonium prior to birth, and then by the time of birth, no more oogonia. So you are not having any more of these by the time you're born. So let's write none by birth. So for this reason, the fertility window for people that make eggs is shorter than for people who make sperm, and that's because you don't have any more stem cells by the time you come out of your mom. So that arrest in prophase 1 lasts until puberty. So everything in the ovary is pretty much dormant until puberty. And then after puberty, with the release of follicle stimulating hormone, primary oocytes complete meiosis 1, which produces a secondary oocyte, and this is the one that's going to become an egg, and the first polar body, and this is functionally a waste cell. So it doesn't get used for reproduction at all. It typically just goes away. It dies. The polar body may or may not divide into 2 additional polar bodies, but again, if it does, they are not for anything. So the secondary oocyte gets about halfway through meiosis 2, but stops in metaphase. So everything is poised at the metaphase plate, waiting for anaphase 2. If a spermatozoan manages to fertilize the secondary oocyte, then meiosis 2 is completed, and the second formal polar body is produced. The result of fertilization is a diploid zygote, which is the first cell of the new individual. So a couple differences between males and females as far as gamete production. In males, it goes on for as long as the testes are active, which is from puberty to death. In females, you gradually run out of eggs until menopause. In males, each individual stem cell reproduction results in 4 active spermatozoa. In females, each oogonium division results in only 1 ovum, which is what we call it when it's finished with meiosis. So very different ways that gametogenesis works between males and females. Also different is the follicular development. So sperm develop in a tube, and ova mature in the ovary, specifically in the stroma of the ovary, which is this dense cell-rich tissue in here. So primary follicles are over here, and these are egg cells with a just developing ring of granulosa cells. So granulosa cells are the helper cells, and they also secrete estrogen. So they're going to help the egg mature and secrete estrogen. And note, for each ovarian cycle, which is what this is showing, so this is actually a time-lapse image. This wouldn't all be going on in the ovary at the same time. So for each ovarian cycle, there is a gradually increasing number of granulosa cells, which causes the level of estrogen to rise over the course of 28 days until ovulation. So a secondary follicle is very large, and now we begin to see an antrum. So antrum is how you spell that. And an antrum is a fluid-filled space. So eventually, the vesicular or Graafian follicle is going to be basically a large cyst on the ovary, and it's also going to be positioned very near the edge. So we have a very large C-shaped antrum. And this is a follicle that is about to rupture. So this is what ovulation looks like. We've got an ovulated oocyte with a corona of granulosa cells, and those will actually stay with the egg as it makes its way into the fallopian tube. And then this is a bunch of colloidal fluid and blood. The cells that are left behind, so these granulosa cells, are going to differentiate into the corpus luteum, which means yellow body, and the yellow body grows larger and larger. It sticks around if pregnancy occurs, and it goes away if not, but the corpus luteum secretes progesterone. And after a while, the corpus luteum is replaced by scar tissue, and that is called a corpus albicans. So here is some ovarian histology for you, and we'll also look at some of this in Histology Guide. The germinal epithelium covers the surface of it, and the cortical stroma of the cortex is a little bit lighter in color than what's in the middle. As you move towards the center of the ovary, it gets darker, and you start to see oocytes. So these big guys are oocytes, and these little cells around the edge, these are the granulosa cells. So primordial follicles have an oocyte with thin, flat, squamous granulosa cells around it. Primary follicles have a cuboidal surrounding cell population. So, primary follicle, here is one. So this is an oocyte arrested in prophase 1. You have 1 to 3 or 4 layers of granulosa cells, usually between 1 and 3. This pink rim is called the zona pellucida, and this is actually not the cell membrane. It's a connective tissue capsule that forces the sperm to burrow through it. This is barely a primordial follicle, and then all of the rest of these kind of pointy cells are thecal cells. Here is a nice secondary follicle, so we do see an antrum. But it's not C-shaped yet, so this is not properly a Graafian or vesicular follicle. Also, there is no neck. So the corona radiata, which is the halo of granulosa cells that surround the egg, is developing, but it doesn't have a little neck piece, as you'll see in the next slide. So this is the corona radiata. Okay, so here's the corona radiata again. The zona pellucida is thickened in the secondary oocyte. And we have this little stalk here, or neck, that connects the corona radiata to the cells of the wall. And another name for that stalk is the cumulus ophorus. So you may see that if you use somebody else's Quizlet, for example. Okay, so I love this picture because it's actually a picture of ovulation in action. So the ovary is literally experiencing a rupture and the egg is jailbreaking out of the side of the ovary. So there is no elaborate duct system as there is in males. It just busts out like a jailbreak. So the ovulation event is caused by a surge of luteinizing hormone. So over from day 0 to day 14 to day 28, if you were to draw the relative amount of luteinizing hormone on a scale, it looks kind of like this. Kicking along, and then, oh my god, that's the LH surge. So that causes ovulation, and then the oocyte is released, and the remaining cells are going to become a corpus luteum, which secretes estrogen, but primarily its job is to secrete progesterone. Okay, this is a uterus. Woohoo, uterus. So we've already been over this, so I'm going to just generalize. The only reason I'm really taking pains to point this out is because the hormones that are released by the ovary influence the state of readiness of the uterus to receive a potential embryo. So you need everything sort of to happen in the correct order and at the appropriate pace. And so the uterus and the ovary are talking to each other a lot to make sure that if a fertilization event occurs, there is somewhere for the embryo to land. So the stratum functionalis is this broad layer here. It's lighter in color and has more stromal space between glands. The stratum basalis is darker purple, and it has lots and lots of glands, so less stromal space between glands. And the myometrium, this has no glands at all, and that's because this is smooth muscle. Okay. So let's go and look at some ovary stuff. So I'm going to go to the ovary that I think is going to have the most follicular diversity. And remember, you have access to this resource too, so you can follow along with me. And oh, look, what a nice diversity of follicles here. So based on the number of almost vesicular follicles here, I'm going to go out on a limb here and say that this is probably not a human ovary. So in humans, the ovaries take turns. One goes and then the other goes, ovulating month to month. In animals that give birth to litters, you have lots and lots of active follicles at the same time. So it's probable that this is a ovary of a litter-bearing creature. But let's zoom way in and look at some different follicles. So I'm going to try and zoom in right here. Hello. Whoa, all right, not quite what I was going for. Let's do it the old-fashioned way, shall we? Okay, so okay, I'm back. I just had to take a short break to go grab my Epic pen, and I've got it here now, so let's go into pen mode. So here we have the germinal epithelium. And then let me... Can I make this skinnier? Yes. I'd like to make that a little bit smaller. Cool. So see these little guys hanging out by the edge? These are primordial follicles, so they have a almost squamous-like surrounding of cells. Here's another one. So these are all primordial follicles. Now let's navigate to somewhere with some more mature follicles, shall we? Oh, this is a good thing to point out. So sometimes, because each follicle is of course a sphere, the slice will not capture the oocyte. So although this particular follicle appears to be empty, it's actually not. It's just that the slice didn't happen to catch the oocyte. Okay, so let's keep moving here. I want to find a nice primary follicle that is good and obvious. Trying to find one that's sort of demonstrative, you know? Need no antrum, because that's all secondary follicle territory. Come on, give me a primary. Well, let's stop here for a second, because this is a nice secondary follicle. So for a secondary follicle, you're going to see a thicker granulosa cell layer. Now I want my dot to be a little bit thicker. And you're also going to see an antrum. This is filled with fluid. And you can see the zona pellucida, which is this thick pink band, as well as the corona radiata, which are the cells that directly surround the oocyte. So this is the corona radiata. So that's a secondary follicle. Okay, so now let's go hunting for a primary follicle. Off we go. I'm going to zoom out a little bit. That'll help me look a little bit better. Yeah, that's a good one. So let's zoom in here. Where did it go? Not what I asked, but thank you. There it is. Okay, so hopefully if I zoom in right here, I will still have it in my field of view. Perfect. Okay, so let's see what qualifies this as being a primary follicle. Step 1, there is no antrum. There is only 2 layers of granulosa cells. And you can see the zona pellucida here, but the primary characteristic is that there are granulosa cells, but there is no antrum. So no fluid-filled space at all. Okay, let's zoom back out and go hunting for a vesicular or Graafian follicle. This, by the way, is the ovarian medulla. You can see there's lots of blood vessels in the center to nourish the developing eggs. And then these little guys in here are going to be corpora albicans, and those are scars on the ovary from past ovulations. So I like this one the best. Let's zoom in on that. And notice also it's very close to the edge of the ovary. So this one down here is probably also a vesicular follicle because it's also close to the edge, but this one does not happen to have an oocyte inside of it that is visible in this slice, so I'm choosing this one instead. So let's zoom in here. So now we've got a nice big C-shaped antrum. And the corona radiata is nice and pronounced. And the stalk is also obvious. And we didn't happen to catch the nucleus in this slice, but you can still see that it is an oocyte. And we've got a nice zona pellucida, that nice pink line that I just covered over with my marker. Okay, so yeah, that's the zona pellucida, and this is going to be a tertiary or Graafian or vesicular follicle. Alrighty, so let's look at what's next. So I'm going to navigate back to the slide box, and we're going to go look at some sperm developing in their tubes. Okay, male reproductive system. Here we go. And testis and epididymis. So let's look at testis. And I'm going to try and find one with nice big tubules so that I can really zoom in well. So hopefully this will do the trick. Okay, great. So within this slide, there is testis and epididymis, so it's important to be careful. This is epididymis. And then all this stuff is testis. And they're separated from each other by connective tissue, which you can see here. So we're going to zoom way the heck in. Maybe let's see if I can just make that happen automatically by pushing on the link. Wonderful. Okay. Oh, this is a good one. You could even see a little spermatozoan right there. So let me get my pen and I'll start pointing stuff out to you. So remember, spermatogonia are going to be near the edge. So this is one, and this is one, and this is one. So they are on the basement membrane. They have a dark nucleus that's often very round. So these are all spermatogonia. You'll find them all around the edge. Let's next look for primary spermatocytes. Remember, these are going to have dark, round nuclei, but not be by the edge. So here's one, and you're seeing a hodgepodge here because the tissue is semi-transparent, so you can see places where you can actually look through a couple different layers. So here's one. Those are primary spermatocytes. Secondary spermatocytes you don't often see because they divide super rapidly, as you can see here. So that's why they're not linked. But you can see a couple here. Remember the Sertoli cells. Whoopsie, there we go. Let's look at some Sertoli cells. So these are cells that are cushioning the sperm, and they're all in and amongst here, so you can occasionally see the nucleus of one. So they have an irregular nucleus with a single prominent nucleolus, and they are of an intermediate color, so they're not abnormally dark or light. So this is one, and this is one, as well as that. So they're large, euchromatic nucleus with a single prominent nucleolus. So that's pretty easy to see. Okay, so that is testis stuff, and here we can see another spermatozoan. These little guys are all going to be, of course, spermatids. Remember, spermatids haven't necessarily undergone spermiogenesis yet, so they are still circular. They undergo spermiogenesis to form a head, midpiece, and tail, and that's what you're seeing in the lumen here. Okay, so that was ovary and testis, which we saved for later. Now, let's see if we can find some cells of Leydig, shall we? Leydig cells. Let's look at cell number 1. Okay, here we have some. So here we have some cells of Leydig. Now remember, I said they're not always triangular, but sometimes they are. So basically the rule is, see this connective tissue capsule that covers the seminiferous tubule? If it's a clump of cells that are outside of the connective tissue capsule, then they are likely Leydig cells. And they're large and round, and they have eosinophilic cytoplasm. So remember, eosinophils from blood cells are the pinkish red ones, and you can see that these are indeed cytoplasmically very pinkish red. So that's your clue. So if you see something that looks like this but it's purple, probably not Leydig cells, but these are a good candidate. So let's delete all that and go see, population of Leydig cells number 2. Yes, so here's some as well. Okay. So the other task at hand, and this is a long lecture, I apologize, there's a lot going on in here, but we also have to talk about, of course, embryology. So let me go back to my PowerPoints, and we will start embryology. Okay, so embryology is how you make a zygote become one of these. So this is the act of fertilization. You can see this is a sperm. Here's the head, midpiece, and the flagellum. And this is the zona pellucida of an egg, which remember is not the egg's cell membrane, but rather a connective tissue barrier. So this is a sperm who has managed to burrow into it, and this is likely the moment of fertilization. So a spermatozoan can survive for between 48 and 72 hours in the female reproductive tract, and it takes about 10 hours to navigate into the ampulla of the fallopian tube where fertilization occurs. So the result of fertilization is a zygote, which is the first cell of a new individual. And in this picture, you can even see, and this is really cool, some nuclear merging. So It looks like a Venn diagram because that is the moment when two halves of DNA are busy combining. Pretty cool, huh? Oops, let me get my... There we go. Okay, so from fertilization to implantation, and oh boy, I sure do like this Epic pen with the PowerPoint. It's much easier to draw with. So this is day 0, fertilization. You can see the sperm. So the sperm actually do have to not only swim in here, but they have to select the correct fallopian tube as well. So some of them will go right and they will just die in there because they didn't go up the right tube. Sorry, guys. So from fertilization to implantation, the egg is moving this direction. So it's bopping along towards the uterus, and it's undergoing a lot of changes as well. So notice the polar bodies are inside of the zona pellucida here. And the zona pellucida is actually going to constrain the size of the early embryo. And that is because during this phase, you get lots of cell division, but the cells reduce in size. So you get more numerous but less large cells. So there's 2-cell stage, and at this point, each of the cells are called blastomeres, and the 4-cell stage. And then starting at about 16 cells, we call it a morula, which is one of my favorite biology words, morula. It sounds like a vampire name, or I don't know, maybe that's just me. I'm a dork. So an advanced morula is a ball of cells, and that actually undergoes something called hatching, where the zona pellucida goes away. So although we are placental mammals, we still technically hatch, which is fun. Isn't that fun? I think so. So after hatching, the cell is called a blastocyst, and this is a hollow ball of cells with an inner cell mass. So the trophoblast is the outer cells, and the inner cell mass is called the embryoblast. All right. So here is a morula. I would call this an early morula because you can still count the cells. So there's about 16 of them, and they look a lot like a raspberry or a blackberry. They have berry-like properties. So here's a blastocyst. So you can see the inner cell mass here, the central space, and the outer cell mass or trophoblast. So once the blastocyst makes contact, assuming the endometrium is prepared, which based on hormone levels it should be, implantation occurs. So the embryo is going to land on the wall of the uterus and begin the process of moving into the endometrial stroma. So the inner cell mass, which is the future embryo, that's why it's called the embryoblast, sits on the surface, and the trophoblast forms that outer ball of cells. The side of the trophoblast that is invaginating into the wall is called the the cytotrophoblast. So that's the one that's going to move inward. And the finger-like projections of that are going to begin invading the endometrial stroma are called the syncytiotrophoblasts. So you can think of those as the rootlets. So it begins to invade and move on in. Once it travels inside, we've got a couple different layers to consider here. So now we've got what used to be one ball with an inner cell mass. Now the inner cell mass has formed an amniotic cavity, and there's a superficial layer and a deep layer. The syncytial trophoblast is this edge stuff, and the cellular trophoblast is in the middle, and that is what connects to the amniotic cavity. Additionally, by this time a yolk sac has formed. So not only did you at one point hatch, like something that comes out of an egg, but also you used to have a yolk sac. And these little guys over here are called chorionic villi. So this is a developing villus. And chorionic villi are precursors to the placenta. So they will eventually become the placenta via their cooperation with the endometrium. Okay, so first you were one cell, and then you were a little raspberry, and then you were a ball of cells, and then you were a sandwich. So this is the sandwich of which I speak. This superficial layer here is the ectoderm. This is one of 3 layers of your former sandwichness. So the ectoderm, mesoderm, and endoderm each become different parts of your body. So the ectoderm is this flat layer of columnar cells. The mesoderm are the cells in the middle, and these are going to be a result of ectodermal cells moving this way and then peeling off and becoming something else. And the endoderm is derived from the yolk sac cells that face the ectoderm. So those are those. So the ectoderm, endoderm, and mesoderm and their ultimate destinations for what tissue types they become, we will talk more about in lecture, but it's pretty amazing to think that all of the different cells of your body started out as in 3 categories. Okay, so moving right along. Gastrulation is the process by which this occurs. So the primitive streak shows up, and then this cell cycling begins. So it's going from a flat sandwich to a more complex folding sandwich. So what we just looked at is the top view and a little bit of the side. This is what it actually looks like. So this is going to be the primitive streak right here, and this right here. And you can see these cells moving down and in, and then these are going to be mesodermal cells here. All right, so about 13 days after fertilization, you can actually see these. So the ectoderm is always going to be the thickest and darkest. The endoderm you can easily find by locating the yolk sac, and then this layer of cells here is that. Whoopsie. And then the ectoderm is going to be this big thick layer here. Okay. So between 21 and 23 days after fertilization, and note this actual size is really small, so it's only about 1 millimeter to 1.5 millimeters, so very tiny. But you can see that the gastrulation process is much more advanced now. So that inward moving of mesodermal cells and the folding of the ectoderm has extended along the entire primitive streak, and now we see the formation of a basic body plan. So note that there is a tube now with an entrance and an exit. This is going to end up becoming the brain and head. This is going to become the thorax and heart. And there's other midline stuff happening in here. So it's all pretty amazing. All right, so about 28 days after fertilization or so, we've got a larger embryo with more recognizable parts. So we've got pharyngeal gill arches and limb buds, and you can see somites, which are blocks of tissue which will become the spinal column. So the embryo begins to receive nutrients and oxygen through the placenta at this point. So here's a nice drawing of what that starts out looking like. So obviously this does not look like one of these, but it's showing you how the chorion, which is the cellular trophoblast and parts of the mesoderm, end up helping to form the wall. And the syncytial trophoblast is going to start invaginating the wall even further and end up forming chorionic villi. So the blastocele is this area in the middle of the yolk sac in total. This is just showing blood supply. So these little coiled guys are blood vessels. So this is showing you early embryonic blood supply. So we can see a tertiary villus, which is a villus that arises from the tissues of the developing embryo. And these villi contain blood vessels that are reaching into the endometrium. And maternal sinusoids are places where maternal blood enters and is in close contact, but not directly touching the fetal circulation. So that's how you keep fetal blood and maternal blood separate. Okay, so the placenta connects the embryo or fetus to the mom and to the wall. So it's a large structure that clings to the wall, and it's functionally a new organ that a woman grows in addition to growing a new human during pregnancy. It's very impressive. And here's a closer up view of the relationship between the fetal vasculature and the maternal vasculature. So areas filled with maternal blood have chorionic villi poking into them, and diffusion carries wastes away and takes oxygen and nutrients to the developing baby. Note also we have a cervical mucus plug, which keeps everything in here, in addition to all of the layers. So this is what a nearly full-term fetus looks like. You can see the head of the infant is about to be pressing on the cervix. Mucus plug is still in place, however. So when the mucus plug is ruptured and amniotic sac is also ruptured, that's the quote-unquote breaking of the water, which permits the infant to begin leaving the birth canal, or excuse me, the womb. So umbilical cord histology looks like a screaming guy. It's very funny. So there are 2 arteries and 1 vein of the umbilical cord. So the arteries are the blue ones. These are carrying wastes and carbon dioxide away from the baby. And there is 1 vein, and that carries oxygen, nutrients, and hormones to the baby. So the artery-vein dichotomy here is also reversed, just like in the pulmonary circuit, because now we're talking about whether blood is being carried to or away from the baby rather than the heart. And that is it for embryology. So we have models in lab of different cell stages, and that's what you're really responsible for. Unfortunately, there is no histology for any of that on the histology guide, so just go with the pictures in your book and in your PowerPoint. So is that, and I will see you guys on Wednesday.